Today we're going to look at DNA replication. And replication is the copying or duplication of DNA. It must happen before mitosis, meiosis, or binary fission in prokaryotes. As you recall, the cell cycle is made up of interphase, which is the longest phase of the cell cycle, and is comprised of three parts, G1, S, and G2. G1 is a growth phase. However, S is called the synthesis phase, and to synthesize means to make, and this is when DNA is copied. The first step in this process involves helicase. Helicase is basically an enzyme. It ends in ASE, and it unzips the DNA. It breaks that hydrogen bond between purines and pyrimidines. And it creates something called a replication fork. So this kind of looks like a weird looking fork. The next step are DNA polymerases, and they basically are escorts. And they bring in individual nucleotides that are floating around the nucleoplasm, and they bond with the replication fork. You can see on the 3 to 5 prime, they're bonding from the inside of the fork and heading out. And on this 3 to 5, they're going from the outside in. They join each other with a hydrogen bond down the middle, and then each of the nucleotides are being joined by covalent bonds. You'll notice that the leading strand leads out from the replication fork, whereas the lagging strand goes into the replication fork. And these little fragments are called Okazaki fragments. The DNA polymerases are eventually released, and we end up with two perfect strands of identical DNA. You notice that we built the strands from 3 to 5. However, we're going to read this DNA molecule on this side from 5 to 3. Basically, the ligase glues those two strands together, and we are completed. One thing about prokaryotes is that they have a ring of DNA, and so they basically make a copy of this. And it breaks open in one area, and it's called an ori, and it goes around in a circle and eventually makes an identical copy. It makes two.